Hello everyone, welcome to our new video that is about the cell wall of gram-positive and gram-negative bacteria. Bacteria are mainly classified into two main categories, gram-positive bacteria and gram-negative bacteria. These categories are based on their cell wall composition and reaction to gram stain test. The gram staining method was developed by Hans Christian Gram that identifies bacteria based upon the reaction of their cell wall to certain dyes and chemicals. The difference between gram-positive and gram-negative bacteria is primarily related to the cell wall composition. Gram-positive bacteria have cell walls composed mostly of a substance unique to bacteria, known as peptidoglycan or murine. These bacteria stain purple after gram staining. The gram-negative bacteria are composed of only a thin layer of peptidoglycan and an outer membrane with a lipopolysaccharide component, which is not found in gram-positive bacteria. Due to this composition, gram-negative bacteria stain red or pink after gram staining. Now let's talk about the cell wall composition of gram-positive bacteria. The cell wall of gram-positive bacteria differs structurally from cell wall of gram-negative bacteria. The primary component of bacterial cell wall is peptidoglycan. Peptidoglycan is a macromolecule composed of sugars and amino acids that are assembled structurally like woven material. The amino sugar component consists of alternating molecules of N-acetylglucosamine and N-acetylmuramic acids. These molecules are cross-linked together by short peptides which help to give peptidoglycan layer strength and provide them a structure. Peptidoglycan provide protection for bacteria and also define its shape. The gram-positive bacteria cell wall has several layers of peptidoglycan. The thick layer of peptidoglycan helps to support the cell membrane and provide a place of attachment for other molecules. The thick layers also enable gram-positive bacteria to retain most of the crystal violet dye during gram staining, to which the gram-positive bacteria appear purple after gram staining. The gram-positive cell wall also contains a chain of teichoic acid that extend from plasma membrane through the peptidoglycan cell wall. Teichoic acid helps some of the gram-positive bacteria to infect other cells and cause diseases. Now let's talk about the cell wall composition of gram-negative bacteria. Like gram-positive bacteria, gram-negative bacteria is also composed of peptidoglycan. However, peptidoglycan layer in gram-negative bacteria is comparatively much thin than gram-positive bacteria. Due to thin layer, the bacteria cannot retain initial crystal violet dye but picks up the pink color of counter-stained safranin during gram staining. The cell wall structure of gram-negative bacteria is much more complex than gram-positive bacteria. Between the plasma membrane and peptidoglycan layer, there is a gel-like matrix called periplasmic space. Unlike gram-positive bacteria, gram-negative bacteria have an outer membrane layer that is external to peptidoglycan cell wall. Membrane proteins, murine, lipoproteins attach to the outer membrane of cell wall. Another unique character of gram-negative bacteria is presence of lipopolysaccharide molecules on outer membrane. Lipopolysaccharide is a large glycolipid complex that protects bacteria from harmful substances in their environment. It is also a bacterial toxin that can cause inflammation and septic shocks in humans if it enters the blood. There are three main components of lipopolysaccharides: lipid a core polysaccharide, and an O-antigen. The lipid A component attaches the lipopolysaccharide to the outer membrane. Attached to the lipid A is a core polysaccharide. It is located between the lipid A component and O-antigen. The O-antigen component is attached to the core polysaccharide and differ between bacterial species. It can also be used to identify specific strain of harmful bacteria. That was all about the difference between cell composition of gram-positive and gram-negative bacteria. Thanks for watching this video. Please follow our channel and click on subscribe button for watching more explanatory videos.